Okay, so welcome back to this next video in which we are discussing high-density lipoproteins. Okay, right. So, we're currently discussing the ABC transporters, of which we're going to see many examples within this video, and we've already seen ABCA1's involvement in transporting cholesterol and lecithin molecules over to the apolipoprotein A1 to produce these nascent discoid or HDL molecules. Okay, right. So we've discussed the structure of ABC transporters. We've discussed the many different types that we have within humans. What we now want to discuss is a model for how they actually work. Now, this has only been studied in any great detail for a single ABC transporter, and that's the multi-drug resistant protein 1, MDR1, also called P-glycoprotein, and also known as the ABC transporter B1. Okay, right, so we're going to look at the mechanism of this transporter, but the mechanism by which it works we can use to understand the other ABC transporters as well. Okay, right, so ABCB1 is an example of a full transporter, so remember it has these two transmembrane domains, which both consist of 6 membrane-spanning alpha helices, okay, and then you have the first nucleotide binding domain between the two transmembrane domains and the second nucleotide binding domain after the second transmembrane domain. Okay, so we've effectively got these two halves of the molecule. The transmembrane domain 1 with the nucleotide binding domain 1 is one half, and the transmembrane domain 2 with the nucleotide binding domain 2, that's the second half. Okay, so I'm going to bear that in mind when I draw this diagram. Okay, so here is the first transmembrane domain. Okay, here. And then dangling down you have this first nucleotide binding domain here. And these two little invaginations that I'm showing within the nucleotide binding domain, these are binding sockets for ATP molecules. Okay, so ATP is the nucleotide which it refers to within the nucleotide binding domain name. Okay, right. Then we have another one of these transmembrane domains, like so. Okay, so this is transmembrane domain 2 here. And then dangling down, you also have nucleotide binding domain 2 here. So I'm just splitting the molecule into this, these two halves, basically, even though it is one whole protein. So remember, ABCB1, or the multi-drug resistant protein 1, is a full transporter on its own. Okay, so it's not a half transporter. Right, so here's how it begins. It begins with the nucleotide binding domains, which I'm going to draw in blue here, unbound to ATP, so they don't have ATP bound to them. Okay, and at the moment, the transmembrane domains are in this inwardly facing state, so they're facing inwards. Okay, so in green here, these are the transmembrane domains, and currently the entire transporter is in the inwardly facing state. Okay, so it's ready now to bind to its solute molecule, which could be a drug molecule, which will be on the cytoplasmic side of the membrane. Okay, right. So what's now going to happen is the molecule which we're going to transport is going to come in and bind to this cytoplasmic binding domain that's made up by the transmembrane domains here. Okay, so this is the molecule that's going to be transported, the solute molecule here. And I think I'll colour in the solute molecule in red. Okay, then when the solute molecule binds like this to the transmembrane domains, what that will trigger is changes in the conformation of the nucleotide binding domains down here. So remember, these are the nucleotide binding domains, whilst these up here are the transmembrane domains. Okay, And now these nucleotide binding domains will gain affinity for ATP. Now for now, I will just denote ATP as a little egg-like shape here. So these little molecules here represent ATP molecules. And we're going to need 2 ATP molecules. Basically, what's going to happen is when these nucleotide binding domains bind ATP, what happens is one ATP molecule is going to sit in between the 2 nucleotide binding domains here, and another is going to sit in between the 2 nucleotide binding domains here. So what's going to happen is both of them are going to dimerize together, and they'll have 2 ATP molecules holding them together. Okay, right. So when the solute molecule binds then, it triggers a conformational change down here in the nucleotide binding domains, and they're going to gain affinity for ATP, so 2 molecules of ATP are going to bind in here. So let me show this. So here are the transmembrane domains again, and nothing has changed up here yet. We haven't transported the molecule across suddenly yet. Okay, here we go. Here are the transmembrane domains, still in the inwardly facing state, and they have the solute molecule attached. Okay, so I'll colour in the solute molecule again in red down here, and then I'll colour in the transmembrane domains in green. Okay, once again. Now, what has changed is that the nucleotide binding domains have now both bound to the ATP molecules, and now they're going to be dimerized together. So let's draw this here. Okay, like so. And you'll have one ATP molecule there, and then another ATP molecule there. Okay, so colouring in the ATP molecules first in vivid purple here. There they are. And they are holding together the 2 nucleotide binding domains, which are now both coloured in blue here. Okay, and are dimerized together. Okay, right. Now, the dimerization of the 2 nucleotide binding domains like this will then trigger a conformational change on the level of the transmembrane domains, so that they move from facing inwards like so to facing outwards. Okay, so let's draw our next picture. Okay, right. So what's now going to change is the transmembrane domains. They're going to start facing outwards. So once again, here is the phospholipid bilayer of the cell membrane. And now what will have happened is that these transporters will now be facing outwards. Okay? So the transmembrane domains change conformation, and they change into what's known as the outwardly facing state, where the ligand molecule is now exposed to the outward side, okay, to the extracellular fluid. Okay? And intracellularly, meanwhile, the nucleotide binding domains are still bound to one another by these 2 ATP molecules. Okay, so down here we have the 2 nucleotide binding domains, which are still dimerized together, and they still have the ATP bound to them. So the ATP has not been hydrolyzed yet. Okay, so in vivid purple, that's the ATP, the 2 molecules of ATP. And in blue here, these are the nucleotide binding domains. Okay, and what colour did we pick for the transmembrane domains? They were in green, so here are the transmembrane domains in green. And then we have the molecule that's being transported across the membrane, and it's being transported out of the cell. So this is exportation, and it now is on the extracellular aspect. Okay, so once the transmembrane domains undergo this conformational change that ends up transporting the molecule from the cytoplasmic side to the extracellular fluid side, the molecule loses affinity for the transmembrane domains and falls off, basically. Now, when this happens, what will happen is you'll then get a conformational change on the level of the nucleotide binding domains, and what will happen is the 2 molecules of ATP will be hydrolyzed. Now let's discuss this in a little bit more detail. Okay, so ATP is short for adenosine triphosphate. Okay, so let's draw a little cartoon for adenosine triphosphate. Previously, we'd just shown it as this egg structure here. Now we're actually going to draw a more in detail cartoon. So basically, to understand what adenosine triphosphate is, you need to understand what adenosine is. adenosine is the name for a ribose sugar, which is a pentameric sugar, like so, with a fifth carbon coming off up here. So not all of the members of this 5-membered ring here are carbons. This is actually an oxygen at the tip. Okay, so you've got 4 carbons within the pentamer, and then the 5th comes off up here. And then to create adenosine, you have bound to this ribose sugar an adenine organic base. Okay, so let's colour this picture in. So we'll colour the adenine organic base in green here, and then we'll colour the ribose sugar in blue here. So when you bind ribose in blue to adenine in green, the combination of those two together is known as adenosine. Okay, so ribose plus adenine is equal to adenosine. Okay, right. So, to create then adenosine triphosphate, ATP, what we need is to add on 3 phosphate groups onto the adenosine molecule. Now, these are added on to the alcohol group that comes off the 5th carbon of the ribose sugar. Okay, and they will be added on via phosphoester links. So the first phosphate will be added on via a phosphoester link. Then you'll link the 2 phosphates together. So one of them will act as the carboxylic acid group and the other will act as the alcohol group, and they'll form a phosphoester link between one another, and then you add on a 3rd one like so. Okay, so here are these 3 phosphate groups. And I've bent them backwards like this because we didn't really have room to draw them going vertically, but it's more conventional to draw them going up in a line. Okay, so the first phosphate group that's added on to the 5th carbon of the ribose sugar is called the alpha phosphate. The second one is called the beta phosphate, and the 3rd one is called the gamma phosphate. Now, when you hydrolyze ATP down to ADP, what you do is you break this bond between the beta phosphate and the gamma phosphate, and what you therefore produce is an adenosine diphosphate molecule. Okay, and this is just adenosine with 2 phosphate groups stuck on it, the alpha and the beta phosphate, like so. So here is our adenine organic base down here, okay, in green. Then we've got our ribose sugar in blue, okay, here. And then coming off the 5th carbon's alcohol group, we then have 2 phosphates, an alpha phosphate and a beta phosphate. Okay, so this molecule here now is adenosine diphosphate, ADP. Okay, and the other thing that you then create is a single phosphate group on its own, and this is called an inorganic phosphate, and it's often abbreviated just as PI, like so. Okay, so what's going to happen is when the ligand molecule breaks off the transmembrane domains, what will happen is these ATP molecules that are bound in between the nucleotide binding domains will be hydrolyzed down to ADP and an inorganic phosphate, and when they're hydrolyzed, they no longer bind the 2 nucleotide binding domains together, so the 2 nucleotide binding domains split apart, and the ADP and inorganic phosphate molecules are given off. Okay, right. So, what then is going to happen is you're going to go to something like so. Okay, so we're showing intermediates here, okay? So at the moment, we're still going to have the transmembrane domains being in the outward state, okay? In reality, as soon as the ATP molecules are hydrolyzed and the nucleotide binding domains fall apart, the transmembrane domains move back into the inwardly facing conformation, but we're showing the intermediate steps, basically. Okay, so here then are our 2 nucleotide binding domains, or at least here's one of them now, and they're now no longer dimerized together. They no longer have ATP binding them together. Okay, and we've given off these ADP molecules and inorganic phosphate molecules, and I'll show the ADP as this bigger lump here. Okay, so this bigger lump that's now being coloured in orange, this is the ADP, and then the little fragment here in pink, that's the phosphate that has come off the ATP to create ADP. Okay, right. So, here are the nucleotide binding domains in blue here that are now separated. And here are our transmembrane domains in green, which are still facing extracellularly but have no transporter molecule attached to them. Okay, so they have no substrate attached to them. And now what will happen is once the nucleotide binding domains have fallen apart like this, then the transmembrane domain will return to the inwardly facing conformation. Okay, right, so this is what it will then return to, and now we're back to square one basically. Okay, what can happen now is another ligand molecule can come and bind to the ligand binding domain on the inwardly facing state of the transporter, and then you can go through the entire cycle again and transport that substrate molecule out of the cell. Okay, so this is how the MDR1, the multi-drug resistance protein 1, can transport molecules out of the cell basically. Okay, and it's very important, as I say, in drug resistance within liver, kidney and colon cancer. Okay, right, so let's think about the stoichiometry here. We transported one molecule out of the cell, okay, and in exchange we had to hydrolyze 2 molecules of ATP. That's a running theme for ABC transporters, that they will transport one molecule across the cell membrane. In this case it was export out of the cell, okay, so this was an exporter. In some cases it will be importers instead, will be bringing molecules into the cell rather than chucking them out. Okay, but the theme is still the same, that we have to hydrolyze 2 molecules of ATP for a single substrate molecule that's going to cross the membrane. Okay, right, so that now finishes our discussion of the ABC transporters. I hope that's been helpful in allowing you to understand them in a bit more detail, because we're going to see a lot of them in the reverse cholesterol transport pathway. Okay, right, in the next video we'll continue on with our discussion of the reverse cholesterol transport pathway.